The aim of this work was to test imprinted gene enrichment in the adult mouse brain using previously published single-cell RNA sequencing datasets. A number of imprinted genes have had brain and behavior phenotypes characterized already, and this has led to the suggestion that imprinted genes are enriched in the brain, but this has not been formally tested. Hence here we have analysed 12 publicly available datasets to question imprinted gene enrichment at three levels of analysis. Level 1 compares expression across a collection of adult tissues. Level 2 compares expression across the brain, grouping cells by region or cell lineage. And Level 3 focuses in on specific brain nuclei to look for consistently enriched cell populations. Enrichment analysis included an overrepresentation analysis and the more conservative gene set enrichment analysis, and this was carried out using a custom list of 119 imprinted genes. Level 1 analysis saw a convergent overrepresentation of imprinted genes in the brain in both datasets, and when PEGs and MEGs were analysed separately, only the PEG showed brain enrichment. And Level 2 analysis saw that across the entire nervous system, neurons were the only cell type overrepresented, and when grouping neurons by their region, the hypothalamus, ventral midbrain, pons, and medulla were overrepresented. Level 3 analysis focused in on these regions. For the hypothalamus, the same key neuronal populations were found to be overrepresented when analysing the neurons of the whole hypothalamus and neurons in the specific nuclei independently. These neurons are associated with pituitary hormone release, feeding, parenting, and circadian regulation. For the midbrain hindbrain, we focus on the monoaminergic system, showing a generalized enrichment in midbrain dopamine neurons and a more pronounced enrichment in serotonin neurons, specifically those neuron subtypes projecting subcortically. To conclude, our analyses show that imprinted genes as a gene set are enriched in the brain, specifically in neurons, specifically neurons in subcortical areas, and even more specifically in behaviorally associated hypothalamic neurons and the monoaminergic system of the midbrain hindbrain.